Welcome back, folks. It's your host, Steele Rogan. Today's episode is packed with two revolutionary topics: meiosis, the wild process that makes you genetically unique, and Mendelian genetics, the OG science of why you inherited your grandma's nose but not her love for polka music. Let's bring in our experts, Dr. Dawson, a meiosis maestro, and Dr. Flores, a mandolin genetics guru. Let's dive in. Dr. Dawson, let's start with meiosis. Walk us through the two-act cell drama.
Sure. Meiosis 1 is where the magic happens. It starts with interphase 1, cells prep by copying DNA, doubling chromosomes, and then prophase 1 kicks in. Homologous chromosomes pair up, swap DNA chunks via crossing over.
Wait, crossing over like a genetic swap meet?
Yeah, exactly. This creates new allelic combos. And then next in metaphase 1, chromosome pairs line up randomly, independent assortment. Imagine shuffling a deck of cards. Each gamete gets a unique hand. Then interphase 1 yanks homologous apart. And then in telophase 1, it splits the cell into two haploid cells.
So why two divisions?
Good question. Meiosis 2 is like mitosis, but without DNA copying, prophase 2 condenses chromosomes. Metaphase 2 lines them up. And then anaphase 2 splits the sister chromatids. Then telophase 2 gives us four unique haploid gametes. Genetic diversity, check.
So you said genetic diversity. Why is that such a big deal?
So there's three reasons. The first one, crossing over, and then independent assortment, and then random fertilization. Without these, we'd all be clones. Sexual reproduction superpower is mixing genes for survival.
Speaking of sexual reproduction, what organisms use it?
Humans, most animals, plants, even fungi. But asexual reproducers like bacteria clone themselves. Asexual reproducers adapt faster. Evolution's a cheat code.
Mitosis versus meiosis. What's the difference?
So mitosis makes identical body cells, one division, diploid clones. Meiosis, two divisions, four unique haploid gametes. Mitosis is a copy-paste. Meiosis is a shuffle play.
Well, thank you. That explains pretty much everything.
Of course.
Welcome, Dr. Flores. Let's talk about Gregor Mendel, the monk who cracked genetics with peas. What's the deal with his laws?
So Mendel noticed traits like flower colors followed patterns. His law of segregation says alleles split during gamete formation. Each gamete gets one allele. Dominant alleles, like purple flowers, mask recessive alleles, like white flowers.
So if I'm heterozygous big P, little p for purple flowers, I'm still purple?
Yep, dominant alleles rule. Only two recessives, like small p, give white flowers. Genotypes, or genes, versus phenotypes, which is the appearance 101.
What about the law of independent assortment?
They're genes for different traits, like flower color, height, which sort independently, unless they're linked. Your eye color doesn't care about your height.
Show us a Punnett square.
Let's do a monohybrid cross. Big T, little t by big T, little t. So this would end up being two big T's. This would end up being big T, little t. This one as well, big T, little t. And then two small t's. So we have two of these, one of the big T, and then one of the small t, small t. So each parent gives either a big T or a little t. The result is 25% big T, 50% of big T, little t, and then 25% of small t, small t. So 75% are going to be tall, these three, and then this one is going to be small or short.
Would you be able to do a dihybrid cross for us?
Sure. So we have all of our letters up here. So this one is going to be big Y, big Y, big R, big R, big Y, big Y, big R, little r, big Y, little y, big R twice, big Y, little y, and then big R, little r. So the same pretty much goes for everything else. So big Y, big Y, big R, little r, big Y, and two little r's. Big Y, little y, big R, little r, then big Y, little y, double small r's. So the same goes for these two rows as well. Big Y, big R twice, big Y, big R, little y twice, and two big R's. Then two little y's, big R, little r. And our last row, big Y, big R, big Y, little y, two small r's, two little y's, one big R, one small r, and then two little y's, two little r's. So this ends up being a 9:2:3:2:1 ratio because of this being 1 and then majority being all of the ones at the top with the big Y's.
So for the viewers and myself, could you explain how you got the top and the side, how you knew what to put there?
So for that, we have to do the FOIL method. So here we have big Y, big R, little r. So in order to get all of these, we had to do the FOIL method. So we had to multiply pretty much big Y by big R, little y by little r, then big Y by little y by big R. So that's how we put all of these in order, keeping the ones with the big Y first into the sections. So big Y, and then we went into little y, little y. So the R, it really didn't matter where it was at. We just had to keep the uppercase Y's first, little y's third and fourth.
How is this used in today's world?
So there's genetic disorder like Huntington's, which is dominant, cystic fibrosis, which is a recessive, breeding mechanisms like drought-resistant crops, labradoodles, and even CRISPR built on Mendel's work. His work lets us understand why some people have dimples, some animals have shorter legs, and why some plants have different colors.
What about Mendel's legacy?
He proved that inheritance isn't random, it's predictable. Modern genetics, all roads lead back to peas.
Mind blown. Thanks to Dr. Dawson and Dr. Flores for decoding meiosis and Mendel. Remember, folks, you're one of a kind genetic remix. Stay curious.